Hello, good morning, good afternoon. I'm Dr. Jeffrey Feld. You can call me Jeff. I'm a marketing and content manager at Curiox Biosystems. We have a really fantastic webinar today featuring Professor Mats Bemark of the University of Gothenburg in Sweden. Dr. Bemark received his PhD at Lund University in Sweden. He did his postdoc at the MRC and the Laboratory of Molecular Biology in Cambridge, UK. He's been at Gothenburg since 2001 and also working at the Sahlgrenska University Hospital. Pardon if I pronounced that wrong. Dr. Bemark is an expert in B-cell biology, particularly working in the gut, but also other parts of the adaptive immune system. He's one of the first 10x genomics chromium system purchasers in Sweden. So he's been working optimizing single-cell genomics, single-cell multiomics for quite some time. And we are really excited that he's been a Laminar Wash customer and user for about a year. That's really helped him enable some of his studies that he's going to talk about today. So before Dr. Bemark will present his experiments and his data, I just wanted to give everybody a quick overview of Curiox and Laminar Wash, just so we're all on the same page. So here we go. We'll switch over. So I'm going to talk about Laminar Wash sample prep methodology, just so you understand, the systems we're working with, and then adding cell hashing and multiomics to your studies to really, expand and save money and save time, make the most out of your samples. And also, he's going to show how Laminar Wash enables some of the studies that he's done. So an antigen-specific immune responses. So these antigen-specific lymphocytes are typically very rare. So using Laminar Wash has really enabled some of these high contrast studies. So we're really living in the age of biology where we have where cells are really the stars. We have samples with cells, so whether they're biopsies or from blood or from cell lines for manufacturing, and we have very advanced techniques for analyzing the contents of those cells, whether that's flow and mass cytometry or single cell sequencing, which will be the focus of today's talk. And these advanced technologies are really enabling this next generation of therapies, vaccines, and even cell-based manufacturing and diagnostics. But the problem is that there's a bottleneck in the process of getting those samples that have cells to those technologies for analysis. And it's really based on an old technology, the centrifuge, that's been around, practically unchanged since the 1950s. So our conventional methods that rely on centrifugation are difficult to automate. So there's a lot of manual movements that are difficult and expensive for robotics to replace humans. And because of that, there's a lot of inter-user variability. So different folks at different locations may flick differently. Their plates, they might extract the supernatant a little bit different in the morning than they do in the afternoon. So you get this inherent variability in these manual methods. And finally, exposing cells to sometimes even thousands of times the force of gravity is disruptive. So Curiox, in particular, using our Laminar Wash technology, is trying to address this bottleneck. So what Laminar Wash offers researchers are options for automation. So that you can essentially speed up the process, make it more hands-off, and make it more reproducible, since you're relying on machinery to do the work rather than manual hands-on techniques and training. And also the method, as I'll demonstrate, relying on laminar flow, is much more gentle on cells, so it keeps them in their native state. So really, we're bringing the sample prep to the 21st century to align with these cool cellular analysis tools and next generation therapies and technologies that we're working on. So real brief, just wanted to talk about how Laminar Wash changes sample prep. So first, as I mentioned, it keeps cells in their natural state. By not exposing them to centrifugation forces, not pelleting them, you end up retaining, you can often retain more cells and keep them in a happier state. So here, I'm just showing you some fluorescence microscopy examples of, these are circulating tumor cells. So this particular example, I think, is pretty relevant for what Dr. Bemark is going to talk about. with cells that are rare, that are difficult to come by. So we're talking about maybe 50 to 100 cells per sample. And you really striking differences here. You can see the blue fluorescence is the nuclei, and red is an endogenous biomarker protein on the surface. And so you're looking up at the centrifugation image here. These cells don't want to be centrifuged. You can tell, right? They look sickly. They aren't filled with liquid. That this marker itself has low fluorescence, and this was actually exposed longer than the contrasting image over here with laminar wash. So these cells look much healthier. They're turbid. The marker, this biomarker has high expression levels. So just the cells are in a state that we can now measure them much more readily in a natural state. Laminar wash also simplifies your workflows. It reduces the hands-on time and inter-operator variability that we're after for 21st century sample prep. So this is just some example data from a time motion study that our research group in Singapore conducted, looking at the differences between manual centrifugation and laminar wash and producing and going through a typical workflow. And these were equivalent workflows doing 3 subsequent centrifugation steps, or the equivalent in laminar wash is 12 cycles, and I'll go over what that means in the next slides. But you can see that the hands-on time is drastically reduced, and even the overall time is much reduced with laminar wash. You end up with a system where you push a button, you walk away, and you enjoy your coffee rather than having to flick and wait for the centrifuge and add more wash and decant the supernatant, et cetera, that we're all used to. And that also goes with facilitating automation. So having walk-away reproducible results is what we're after, right? And these are some recent data collected with our collaborators over at Charles River using our fully automated system, the Auto 1000. So here we're looking at humanized mouse blood samples, and we're looking at live cells and then subpopulations of lymphocytes. So when we're looking at these in terms of percent frequency from flow cytometry, from gating strategies, so you can see that overall, the numbers of cells, the frequency of cells is really unchanged between the two methods. They don't really affect the, what cells you're actually measuring, but the reproducibility is drastically affected, reduced with laminar wash. So, with live cells, with the overall live cells, we get relatively equivalent reproducibility. But as we get into these subtypes, so your CD45, CD3 cells, 25% CV with the manual method versus, well under 10% for the laminar wash method. And then the CD45 cells without the CD3 marker, again, over 10% CVs for the manual method versus under 4% with the laminar wash method. And this is a walk-away method for laminar wash. So, really getting to that inter-operator, inter-geography reproducibility that we need for advanced therapies. And so here I'm just going to show you a couple of videos explaining how the laminar wash technology works. So it first starts with a unique plate design. We have these hydrophilic surfaces that essentially generate the wells, so that's where your liquid can accumulate. And then we have this hydrophobic surface that replaces the wall in your typical microplate. So you have 96 wells in the plate, and those hydrophobic layers are going to allow those droplets to form. So you can just use normal pipetting, whether it's robotics or an 8 or 12 channel pipette, to add your cell samples to the plate as you normally would. And these wells can accommodate about up to 80 microliters of cell sample, and which you can increase using a large volume adapter if you need to handle larger volumes. And you can actually add up to about 10 million cells, or well down to even 1 cell per sample, if that's what you're looking at, say like your circulating tumor cells. The next step is we're going to allow those cells to gently settle naturally by gravity. So instead of subjecting them to centrifugation forces, they will naturally settle to the bottom of the well. And the hydrophilic surface actually, cells don't adhere to it. They don't interact with the surface. They just rest comfortably on the bottom. And you combine this step with your antibody binding step, 15 to 30 minutes, depending on how many cells you're adding to the sample, and allow your cells, of course, to bind as they're settling. What ends up in the bulk solution is your unbound antibodies or reagents, your dead cells, and your debris. They're going to float and not settle to the bottom of the plate. So as opposed to centrifugation, where all that extra, for lack of a better word, garbage, you're pelleting it to the bottom of your tube or your plate. And then next, we combine our plate with the instrumentation. So here we're showing you our HT2000 instrument that has a touchscreen and handles 96 wells at a time. So here we have 2 nozzles. So first, you select the parameters for washing, which I'll go over in a second. But each well has 2 nozzles, so you end up with 192 nozzles total. You have one that delivers liquid on the left and one that's aspirating liquid on the right. So you can see as you're adding liquid to the drop, you're creating a laminar flow that removes all of this unbound reagent, antibody, dead cells, debris, and then the aspirating nozzle is going to collect that and remove it to waste. And down at the bottom, the laminar flow is essentially, the current is essentially 0. So you have your cells unaffected by the flow. They're resting comfortably bound to their antibodies as you're removing that excess, garbage that you don't want bound. The user can select how many wash cycles they want. So each subsequent cycle essentially is just, a longer walkaway step, but can give you that extensive washing that you're after. And then you can also control the velocity of the wash to, essentially mitigate cell loss or increase the speed of your wash cycle here, and really just optimize exactly what you need for your application. And so lastly, just before I turn it over to Dr. Bemark, I wanted to just go over, some of the what Laminar Wash can do for your single cell sequencing studies. So first, let's go over just some of the problems that you experience. Typically, we hear about from customers and researchers in single cell with the sample prep process. So first, it's difficult to remove debris from dirty samples. So if you're collecting, say, a biopsy for a tumor microenvironment study, there's just a lot of stuff you got to get rid of, and it's difficult to do so with the centrifuge. And then we're talking about retaining viable rare cell types. For example, peripheral B cells are in low abundance, even in blood. So you want to retain those cells for studies, and that can be an issue as well. And then finally, for doing some of these multiplexing studies that Dr. Bemark is going to focus on today, so cell hashing multiomics, you want to remove a lot of that unbound antibody to get better signal to noise. And I'll let Mats go over that in more detail. So what are the answers to these problems with conventional washing? Essentially, they're centrifuge more, wash, wash, wash as many times as you can. So you end up with an unwieldy number of centrifuge-based wash cycles. So for example, 10x Genomics recently released the CellPlex method, a great method for using lipids for cell hashing, but there are 5 individual wash steps where you're going to the centrifuge and washing your cells. And each one of these spins invites additional cell loss and variability amongst users. So, there's got to be a better way, right, rather than this old centrifuge method. So what does Laminar Wash offer? First, those debris, dead cells, and unbound antibodies, they float, right? They float in they remain suspended in the bulk solution, and you remove them by laminar flow. And you're able to achieve thorough washing essentially by just increasing the number of wash cycles and enjoying your coffee a little longer. So there's no additional hands-on steps. You don't have to go to the centrifuge 5 times. You just click 25 cycles, as BioLegend recommends for doing TotalSeq using Laminar Wash. So 25 cycles on Laminar Wash takes somewhere between around 8 or so minutes versus the 5 trips to the centrifuge to get essentially the same, if not better, cleaning using Laminar Wash. So with that, I'd like to hand it over to Dr. Bemark, who's going to go over more of the biology behind the single cell sequencing and some of his immunology studies that he's using Laminar Wash. So with that, I'll turn it over.
So, welcome. I'm sitting here and starting to get towards the evening here. I guess that's different from most of you. Just see here. And now you see my main screen, yes. So I would like to discuss today is a bit how we use Laminar Wash and how that enables us to use single cell multiomics and cell hashing on very small samples. So just to sort of remind you, and I think that most of you are quite aware of this already, how drop-based single-cell RNA-seq works. That you have these microbead with tags on them, where you have specific cell barcodes. So each of these microbeads have their own barcode. And by then mixing them with cells, we end up in compartments, small droplets, where one of these microbeads sits with one cell. And when we lyse the cell, that will release the mRNA. And the mRNA is then labeled by using this as a target for either the first strand or second strand synthesis. And based on that, you then sequence the data on one of these major sequences, usually from Illumina. And the data that you then will actually get out from an experiment like that is like an enormous spreadsheet, where each of the barcodes here represent then one of these compartments where you have a cell and a bead, and you have all the genes in your experiments. And for each of these barcodes, you will then know if it's expressed 1 copy or 0 copies of gene 1, gene 2, and so on until gene N. And when we present the data from those types of experiments, we usually show them in these types of 2D projections, where you then can see each of these dots represent a single barcode, that is a single cell. And then if two of the cells are similar to each other, they will end up close to each other. So for example, barcode 1 here, 045, will be close to barcode N, which is 055, whereas barcode 2 will be in a different place on the diagram. And of course, this reminds you a little bit about the typical assays that immunologists use, which is flow cytometry, which also is a method for analyzing single cell, and it's probably the single cell method that is one of the most used single cell methods around, and also one of the more powerful applications for single cells. And essentially in this case, you label your cells instead with antibodies against cell surface markers or cell surface proteins. Can also be labeled intracellularly, but mostly you label on the cell surface. These have different types of fluorochrome on them, and then you let the cells pass through a flow cell where they are enlightened by a laser light, and the laser light will then activate the fluorochromes, and you will then be able to measure how much antibody that is bound to that cell. And usually you present that also in dot plots, but these are then directly correlating with the expression of antibody 1 and antibody 2, for example. So here we can see one population that expresses both protein 1 binding to antibody 1 and protein 2 binding to antibody 2. And that is probably the favorite method for immunologists to define cells. However, when comparing these methods, it has to be remembered that these are actually very different types of experiments. So the first thing is that it's very different from looking at defined proteins, in this case, maybe in the order of up to at most 25 proteins that you have defined beforehand, versus looking at all transcripts in a single cell. And of course, that requires different types of analysis. The other one thing that has to be remembered, of course, is that it's a difference between transcriptome versus the post-translational modifications to proteins that you actually measure with flow cytometry. And also, what has to be remembered, which is one of the features that you usually stumble on when you first encounter single cell analysis as an immunologist, is that the read levels are rather low. So usually when you look for genes that are expressed on in a single cell, you just look at maybe 5,000 transcripts, and for genes then that are expressed in less than 1 in 5,000 RNA transcripts, they will actually not be detected in every cell that expresses it. Whereas for flow cytometry, we usually sort of define something as positive or negative depending if we actually detect it. There's also very large variations in the number of cells that you can analyze. On a modern flow cytometer, you can add up to 5 million cells in one single experiment, whereas for single-cell RNA-seq, for reasons that I will come into later, usually you don't add more than maybe 5,000 cells in each experiment. There are also very large differences in the prices of these types of experiments, where one experiment with single-cell RNA-seq may cost you up to maybe $10,000 or more, whereas one single flow cytometry experiment just costs in the order of $100. When it comes to rare cell analysis, I just wanted to show you this as a reminder of the immunological way of thinking, or the adaptive immunology way of thinking, where you start with cells. And when you create your lymphocytes, both the B and T lymphocytes, you actually create diversity. So each of the cells that is created in bone marrow and thymus and leaf are called naive cells, and each of them can be defined as having one specific receptor that actually is created through DNA recombination during development. So if we now look for different cells here, all the different T lymphocytes will have different receptors, and all the different B lymphocytes will have different receptors. And what happens during an infection, such as SARS, for example, is that some rare cells of the naive cells will recognize that antigen. That antigen will activate that particular cell, and the cell will start to proliferate. So the proportion of cells now recognizing that antigen will increase by cell division. And that means that these are all the same. They are actually exactly the same when it comes to the antigen receptor. But they still make up very small proportion of all cells, and that is even more true when we go over to a memory response, where we sort of keep some of these cells that have been activated once before, but these cells are now very few if you count the number of naive cells. So that means that if we want to look for these antigen-specific cells, the ones that were involved in a response here, they will be a very small proportion of all B cells or all T cells at any given time. And how can you then detect these very rare cells? And I just want, again, to remind you, I think that most of you are very well aware of this, but remind you that T cells don't see antigen floating around. They actually see peptides sitting in MHC pockets on antigen-presenting cells. And it is then the specific peptide sitting in that pocket that is recognized, so that if a different peptide is sitting in the pocket, that will not be detected by that T cell. And B cells, on the other hand, detect antigen floating around. By binding to that antigen, can recognize that antigen. And for detection, we then need different types of reagents for T cells and B cells. So for the T cells, we need what is known as MHC multimers, where we have MHC1 or MHC2 molecules sitting with peptides. And these peptides will then be able to bind to the T cell receptors. And if we then have a detection reagent, for example, a fluorochrome or a DNA tag, we will be able to detect that the cell is now binding to this specific MHC multimer. For B cells, on the other hand, we usually use then antigens, and also for reason of getting higher specificity in the binding, we often link these antigens, again, with some kind of detection to be able to see the specific cell. And I now will bring you into the wonderful world of gut immunology. So I've been interested in gut immunology for a long time, and gut is different from systemic immunity, and it is that has given rise to the idea of the mucosal immune system. So the mucosal immune system is made up of these lymph node-like structures known as Peyer's patches in the small intestine or colonic patches in the large intestine, where you actually have lymph structures sitting just under the epithelium being able to interact with antigens. And at the same time, we have lymph nodes that actually drain the gut, so that from the small intestine and from the large intestine going to the mesenteric lymph nodes, we will have a flow both from the Peyer's patches but also from the gut tissue to mesenteric lymph nodes. So it's general thought that antigen-specific responses to gut antigens will be initiated primarily in the Peyer's patches, but also in the mesenteric lymph node. We have now used one of these MHC tetramers for an antigen from cholera toxin, where we look in mice that have been immunized 1, 2, or 3 times with cholera toxin, and we're looking at the cells that now bind to this. And as you can see, before immunization, very, very few of the T cells from wild type non-immunized Peyer's patch will bind to this specific reagent. Whereas if you then immunize the mice, you will be able to detect antigen-specific cells bind to the tetramer. And this will increase over time, but still the number of these antigen-specific cells will be very low, in the order of 0.25% of all the cells at best, and even lower in the mesenteric lymph node. So the background of non-specific cells is very high still in the Peyer's patch and in the mesenteric lymph node if we isolate B cells or T cells. And this is of course a problem. If we now would like to do a single cell RNA-seq experiment, we can quickly calculate that if we load 5,000 T cells and 0.2% of those actually bind to our antigen, then that means that we only will have 10 cells that are antigen specific, and it will actually be very hard for us to determine which of the 5,000 cells that are the antigen specific cells that we're looking for. Furthermore, even if we manage, the cost for each cell, so to say, in the analysis would be in the order of $1,000 for each cells we analyze. So on the other hand, if we now start with one mouse, we know that we could probably detect in the order of 1,000 of those T cells in all the Peyer's patches or in the mesenteric lymph nodes. And that would of course make the analysis much more efficient for us. We know that we sorted the right cells, and we know that. But as pointed out here before, in the recommended workflow for single cell RNA-seq, the cells are spun down several times, and they have to be counted manually to load the right number of cells. And since one of the biggest disadvantages of using centrifugation is that we actually lose large number of cells in each step, it will be very hard for us to generate sufficient numbers of cells to have 5,000 antigen-specific cells at the end. And also that means that if we start with a very large number of mice to generate those cells, it will be very hard to know which animal each cell came from, and if that was sort of cells that have been proliferating and forming clones, or if that happens. So we set up, when we started using our 10x machine a couple of years ago, we thought a lot about this, and we realized that we needed to be able to start analysis more rapidly, so to say, without spinning down cells. And what we realized was that if we were using our FACS-ARIA sorter, the droplet size, when we used a nozzle of 85, which is a good nozzle for lymphocytes, is actually in the order of 4 nanoliters. And that meant that if we took approximately 40 microliter, that would contain 10,000 cells. And with a little bit of cell death, and that you load in the order of 7,000 to 8,000 cells in a typical single cell experiment to get the 5,000 actually analyzed cells, that meant that if we then directly sorted into an Eppendorf tube cells, we could actually generate cells in a concentration that was ideal for just adding to the single cell experiments. However, we also were a bit concerned. So first we actually only used PBS, but we also now know that the BD sheet fluid works just fine for the liquid that is around the cells when we sort. And it's also very, very important to pre-coat these cell tubes and add a little bit of fetal calf serum to make sure that the cell's not sticking to the cell walls, because otherwise you end up with essentially no cells because they stick to the plastic here when you work with such low number of cells. And using this method, we then generated T cell analysis from both total T cells from both Peyer's patches and mesenteric lymph node, or for the antigen-specific T cells from these. And this, I will not go into the actual data in any detail, but it's in a submitted manuscript that we're now waiting for making a revision to. And essentially what we have seen is being able to then analyze the cells. So we first used all of the cells that were in the Peyer's patches and mesenteric lymph node to make a map of cells that were in mucosal tissues. And then we could then map the antigen-specific cell and asking to which populations of these they primarily belonged when they were activated by cholera toxin. Very similar methods have been used in other studies that we have been performing. So this was a collaboration with David Angeletti, where we helped to set up exactly the same method for B cells, and where he then used the hemagglutinin part of the influenza virus to sort antigen-specific cells from several different time points and several different organs, and performed sorting of these specific cells. And it was recently published in Cell Reports. And another interesting study that was a bit fun to do was that we collaborated with Patricia Mendoza Garcia in the laboratory of Ruth Palmer, who works on Drosophila, where we then took Drosophila embryos. Of course, we needed several embryos to do this. We prepared single cell solutions, and we actually FACS sorted those single cell solutions after staining them with a live dead staining. And we managed to do single cell RNA-seq on those cells, both from the whole embryos and maybe more importantly, from embryos where we had GFP expression in a certain cell type along the gut, part of the gut here, where we could then define subtypes of cells among the green GFP expressing cells in the embryos. And that's also under revision at the moment. But this is very fine, and it worked when we maybe had 10,000 cells from an organ or from an experiment. But if we now wanted to look at more different donors, organs, time points, that will become very expensive, since each time we loaded our 10,000 cells, that would cost us in the order of $10,000. And also, if we now wanted to look at very rare cells, so for example, the antigen-specific cells from one specific organ, from one specific time point, and not mixing it with other cells from other organs, should we end up then with just using 100 cells for a single cell RNA-seq experiment? Or how should we do it? This would be very expensive. And we also would like to be able to compare the data that we got from the single cell experiments more easily to our flows experience, so that when we define a specific subpopulation of cells using transcriptomic, we know what population that would be in a flow cytometer. And of course, we cannot really do that because transcriptomics and binding of antibodies to proteins are at the different ends, so to say, of gene regulation, and may not always follow each other very robustly. so that there could be that the one cell has turned off a gene while a specific antigen is still expressed, or that there are post-translational modifications or splicing going on that would make it very hard to detect by our single cell setup, but would still be able to detect with antibodies. And as you probably are aware, there has been a development of assays for this more recently, which is called cytoSeq or REAP-seq, which essentially is based on the principle that you have antibodies, but instead of fluorochromes, you will have a DNA strand attached to these antibodies, which will contain both a part that you can be using for generating a second strand transcript and a specific barcode that would then be unique for this particular antibody. So that, for example, CD19 has one barcode, CD45 has a different barcode, and CD3 has a third barcode, and so on. And the principle here would be that if you then have a cell with that antibody bound to the cell surface, when this is lysed in the compartment for single cell analysis, it will also release the antibody with the antibody tag, meaning that both mRNA and the antibody oligo will generate transcripts that can be sequenced. And in this way, we will actually generate a data sheet that looks first for the transcriptomic data and then at the number of antibodies that actually was binding to the single cell. And these will be linked by the barcode, because the cell barcode is on the bead and there is still just one single bead in that compartment. And this was first prescribed by Stöckius in Nature Methods. There was also another paper for REAP-seq that was very similar in time. Another method that also was then developed using exactly the same principle was also developed by Stöckius in the lab of Satea. And that was based on, instead of using different barcodes for different antibodies, you can find the same antibodies binding to some common antigen with different barcodes on them. So that if you now had sample A, you had one barcode on that antibody, and if you then took sample B here, you will have an antibody binding to the same antigen but with a different barcode, and sample C here is a third antibody binding to the same antigen but with a different barcode. And that means that after labeling those cells, you can actually pool them. And when they're pooled, if you now do the single cell RNA-seq based on the barcodes, you can go back after doing the transcriptomic analysis and look that these cells were the blue cells from sample 1, these are the red cells from sample 2, and these are the green cells from sample 3. And this, of course, enables you to label cells, and if you have very few cells, then mix cells to be able to analyze things that were too expensive or too hard to analyze if you just had, for example, 500 cells or even less. And the reason why this works is not any magic or anything like that. So hashing works because when you do a DropSeq sequence analysis, what happens is actually that you don't have, the reason why you can just use 5,000 cells is not that you just have 5,000 compartments. You actually have beads in approximately 100,000 compartments. And you then randomly distribute the cells over those compartments. And that means that if you use now 5,000 cells, those 5,000 cells will primarily be alone in their compartments. So essentially, if one of 20, so if we use 1/20th of the cells, essentially they will fall in 1/20th of the compartments. And thinking what will happen and how likely it will be that if we add one more cell, how likely that will be to actually fall in an empty compartment or in a compartment already occupied by a cell. It will be in the order of 1 in 20 of the compartments that will be doublets. So the doublet rate is really a measurement of approximately how many or how large part of the total number of compartments that are filled with cells. If we now add a second sample with cells, you could imagine that, well, that may increase the levels of doublets that we cannot, and so that the risk will be that we increase the number of doublets. And the first thing that is obvious, since we add these 5,000 cells to this already largely empty compartment space, most of them will actually fall into their own bubbles without having any of the other cells in that compartment. Some of them will generate doublets, exactly like we did with the first sample. Approximately 1 in 20 will generate a doublet, which we cannot detect based on just looking for anything in that compartment. Because if two cells end up in the same compartment, they of course will be sharing the barcodes. However, we also generate 1 in 20 cross-hash doublets, like this blue one here that contains both one of the yellow ones and one of the green ones. In this case, we can actually very easily remove these doublets, because if there is a doublet, they will actually contain 2 different hashtags, 2 different hashing antibodies, and in that way we will be easily remove that cell from the analysis. So the actual doublet rate will not increase particularly much from sort of background in the 2 different samples by mixing 2 different samples that has hashtags on them before mixing. And when these 2 methods were developed, luckily for us and for everybody else that would like to do those types of experiments, BioLegend very early on decided that they wanted to increase the number of antibodies available for this type of experiments. So they have released both hashtags and antibody against different targets on cells with different cell barcodes on them. And at the moment, they have 3 different systems. One that is general and can be used in any type of dropSeq. One that is specifically developed for 10x genomics 3' system, and one which we will be using, which is based on the 10x genomics 5' immune profiling system. And for each of these, they develop both from human and from mouse 10 different hashtags, 15 actually for the first system, and they developed in the order of 200 to 300 different antibodies against human antigens, and between 150 and 200 antibodies against mouse antigens for each of these labeling systems. So this now actually allows us to do very easily by ordering antibodies from, we don't have to develop them ourselves, we just order them from BioLegend with different hashtags or different antibody specificities. However, there are still things to be considered here. And one is that the expression levels of cell surface antigens is very variable, and I will come back to that. And for this reason, it's actually so that you, in single cell experiments, actually will aim to use subsaturating levels of antibodies, which is very different from working in a flow cytometry, where you usually always try to reach saturation of binding to get the best ability to differentiate between bound antibodies and non-bound antibodies. And you also need to be much more careful when you wash yourself, because, and I will also discuss this, why it's so important to very extensively wash your cells when you use them for single cell RNA-seq analysis. Actually much more important than for flow cytometry. And finally, of course, for us, the big culprit here is how can we do this for very small numbers of cells? Because we cannot really spin down cells in these very small amounts several times to wash them. And there is, of course, when we started using the system from Curiox with the Laminar Wash method. So first thing I wanted to point out is that cell surface antigens are expressed in very different levels, with, for especially the CD45 antigens, being expressed in very high numbers, in the order of maybe 200 to 300,000 of those expressed on every single cell. They actually make up a calculated approximately 10% of all surface antigens that are on the cell. On the other hand, other antigens may be expressed in much, much lower levels, less than 1,000 per cell. And then that means that in this case, if you actually saturated the binding here, in this case, you would find almost 1,000-fold, but at least 100-fold more transcript from antibodies binding to CD45 than from antibodies binding to CD3, for example. And this can actually be appreciated on flow cytometry plots, because if you look carefully on flow cytometry plots, they're almost always presented as log scales. And that is because of the very large difference. So here we look for B220, which actually is a CD45 antigen. And you can see here that the difference between the background and the positively stained cells is several thousand-folds in increase in fluorochrome intensity on those cells. Whereas other antigens, such as CD93 here, for example, the difference between the non-stained cells and the stained cell is much less, maybe just 10-fold or so. And this, of course, means that the difference will also be very different if we now use the antibodies. And for this reason, when flow cytometrists try to calculate the amount of antibody they're using, they usually do what is known as a titration of the antibody, and you look for having the highest possible concentration that will give negative results for your negative cells and will give as high expression levels as possible for your positive cells. So in the case of CD11C here, for example, already at 0.5 here, we will reach saturation and we will not sort of get better resolution by adding more antibody. Whereas for other antigens, such as CD45, by adding more and more antibody, we'll get better and better separation between the negatives and the positives. However, if we did that for the cytoSeq experiment, used saturating amounts, the problem here would be that if we saturate CD45 binding, for example, that would actually mean that 1/10 of all sequencing done for the antibodies will detect CD45. And here we have another of these very strong antigens, CD54, and in this experiment, using saturating amounts of antibody at BioLegend, they found that that actually generated almost 10% of the reads from antibodies were for CD54, the barcode for CD54. And after then, instead, diluting that antibody to a much lower level, then they could end up with numbers that were more similar here, 0.86%, that other lowly expressed antigens reach. So a bit counterintuitively, the more highly expressed the antigen, the less antibody you should be using in a cytoSeq experiment to allow for the sequencing to actually detect as many as possible of all the antibodies expressed on the cell surface. So next I sort of wanted to point out was the problem of free antibodies. And for flow cytometry, of course, we wash our cells, but usually people are not very, very careful when you wash the cells. You want to remove most of the free antibodies, but it's not that critical to actually remove every single antibody. And the reason for this is, of course, if you have a perfectly washed sample coming in the stream, you will have these collections of antibodies sitting on the cell surface, very high local concentration of antibody. And if we then would have some antibodies that are free antibodies floating between the cells, that will not generate very much noise at all. So the problem here, if you have some free antibodies in flow cytometry, it's not a major problem. And in clinical flow cytometry, you actually often not even washing the cells after staining them, and it still works well for most antigens. However, for CytSeq, it's a very different situation. Here, the actual volume of the cell within the droplet is very, very small. So in the case that you wash the samples very carefully so you don't have any free antibodies, of course you will then only have the antibody oligos and the mRNA in the droplet. And in the empty droplets, the ones without cells, you will don't have neither antibody nor mRNA, and you will not generate any transcripts. However, if we now see what happens, even if you have a little bit of free antibody, since the volume of the droplet is rather large compared to the cell, even if you have got a low concentrations of antibodies, you will see that these free antibodies will end up both in the droplets with the cells and in the droplets without the cells, meaning that the antibodies now will give very large background in the experiments, and meaning that you will be hard to actually detect antibodies that are expressed at low levels on the cells, since there will be so many free antibodies already in the sample. And of course, this was where we started to get interested in the QRX system. So the QRX system here would allow us now to, instead of trying to spin down 10,000 cells repeated times, and anybody who tried to spin down 10,000 cells and wash them repeated times knows that after 3 to 4 washes, there are no cells, maybe 500 or something like that, left after all the washing steps. We then thought that this laminar wash method should work well, even for minute numbers of cells, such as 10,000 cells or even less, for staining cells with antibodies. So I will now present 2 different experiments we've done with this system where it has worked very well for us. And the first one is, again, in the gut. We're now looking for antigen-specific B cells instead of antigen-specific T cells. We use cholera toxin, but now with a haptin NP coupled to it. And we then take antigen-specific B cells from a transgenic mouse, transfer them into a wild-type mouse, and orally immunize these mice. And then, of course, the antigen-specific cells that are specific for MP will be activated by the oral immunization, and we will see antigen-specific cells being very highly expressing GFP, making them very easy to sort for further experiments and analyzing. We have used this system extensively before and looked for antigen-specific cells within the phase patches, where we can see them primarily in the germinal centers, but also to some extent up in the subepithelial dome. And here, in collaboration with Siv Schulman at the Weizmann Institute and Adi Biram, a very talented PhD student of his, we can now see the antigen-specific cells. This a bit counterintuitively labeled red here, but you can see the antigen-specific cells up in the said region, the subepithelial dome, as well in the germinal centers, as well as cells traveling between the 2 compartments of the Peyer's patch. So using this system, we now set up to do an experiment where we actually analyze cells. That's 4 different days after immunization. Day 5 is the first day that we really can detect any immune response, until day 18, when we already have a very much declining immune response. And we wanted to look in 4 different types of organs. We wanted to look in the spleen, the mesenteric lymph node, the proximal base package, the ones that are closest to the stomach, and the ones that are distal from the stomach. And we wanted to compare the amount of response in these different organs. And ideally, we also wanted 3 individual mice from each time point. And if you do the calculations here, we realized that that was 48 different organs that should be analyzed. And if we could done this as a normal experiment, it would have cost us in the order of $500,000 just to do the single-cell RNA-seq on those cells that we needed from the different organs. So by using cell hashing and CytSeq, we then instead started with 2 mice. We always took 1 mice where we expected less cells, for the example, the day 5, and 1 mouse with more cells, which example would be the day 8 mouse. We then isolated the cells individually from the different organs, and we then did cell hashing before pooling all the cells. So the cell hashing and washing of the cells after hashtagging was done individually for all the different organs. But we then pooled the cells, sorted the GFP positive cells into a sack like we did before, into an epidraft tube. We usually started with this fewer cells. We usually got about 5,000 cells in total from 1 mouse. And we then added cells to the same sorting tube from the second mouse that then was using different hashtags. And we then labeled them with 37 different CytSeq antibodies and also washed it on a Curex system that we were using for removing unbound antibodies. And to make the story very short, the hashtagging worked very well. We had here, for example, the PP proximal, PP distal hashtags, MLN hashtags, spleen hashtags. As you can see, it's very easy to see which different cells you can see with a different hashtag. If we then put these cells, so these are now the transcriptomic map of the cells, you can see that the PP proximal and PP distal cells were actually distributing quite evenly over these different cell types that you encounter during an immune response, whereas the splenic cells, where we did not really didn't expect any real response, but rather just cells moving into the spleen as plasma cells or as memory cells, we could see very clearly that we had the memory type cells here and plasma cells over here. And using the hashing, we could also then build an overview over time and organ. So here you see the day 5 cells that we isolated, the day 8 cells, and also measuring the different. And you can see how we clearly then end up with this splenic response after 18 days, while you have this very early response made up of totally different cells early in the response. Using the CytSeq, we also are able to very clearly define the different cell populations. The cells that we isolated were clearly B cells. They expressed B220, a typical B cell marker, except for this one, which is the blast plasma cells, which is expected to have much lower expression of B220. We also saw that IgD was expressed over here among some of the more memory-like cells, and that CD38, a marker that is downregulated in germinal centers and on activated B cells, were clearly only expressed at these cells that were more memory-like, whereas CD138, a typical plasma cell marker, were expressed over here in what we thought were the plasma cell population. So the antibodies from the CytSeq clearly working, even though we labeled them only very late in the experiment when we actually did after we had sorted the cells and when we only had in the order of 15 to 20,000 cells, the CytSeq was working. And of course, this is a continuing work to analyze all the data that we have from this experiment. A different experiment that also has been performed during the last month or so is a COVID project where we had patient samples frozen down. We had taken PBMCs, frozen down the PBMCs, and we had samples from quite severely sick people ending up in hospital, where we took 2 to 3 samples while at hospital, and also samples 3 months after recovery, 6 months after recovery, and 12 months after recovery. And we then again, for each of these samples, wanted to have 3 different populations. We wanted the PBMCs, straight from the frozen cells. We isolated B cells by depleting other cell types using a stem cell kit, and that became #2. And we then labeled them with antigen, with markers on them, and cell sorted, as we have done before, for getting the antigen-specific B cells out. And the setup was very similar to the last time. We started to isolate the cells, we hashtagged them, we then pooled them, we added them onto the TotalSeq, onto a chip from Curiox for staining, and we now used from BioLegend a TotalSeq universal cocktail that contains in the order of 130 distinct antibody specificities. And we also added some additional antibodies. I think it was about 5 to 10 additional antibodies that were not in the cocktail, but that interested us in B cells and T cells from the severely sick people. I just want to mention that for the B cell antigens, we used a system from Immudex, which is based on that you have ClickMir, that's called a decode ClickMir, where you have both streptavidin sites where you can bind your biotinylated antigens, which we then bought biotinylated S protein and RBD and N protein from the coronavirus. And we also had then the DNA barcode as well as a PE labeling, making it possible both to sort the cells first based on the binding, but also to detect antigen binding on B cells based on this tag. And for the 6 different patients, you can look at all that they essentially healthy patients having most of the populations that you expect from that type of analysis, and also that most of the cell types actually are quite similar even during hospitalization. There aren't that big differences actually in the general cell populations compared to recovery, where you can see that there are some cell types here. For example, we have the plasmablast, but also some cell types down here that are clearly enriched in the patients during active disease. We could compare transcriptomics and antibody binding. And here, for example, you see the transcriptomics for CD19 compared to the binding of antibody against CD19. Very good correlation, as you can see. We can also look for major cell types very easily identified. So here you can see that the T cells are over here to the left from the PBMC. You can see that the CD4s are primarily down here, while the CD8s are either up here or down at the very top here. You can see the B cells, as we already discussed, NK cells up in this blob expressing CD16, and dendritic cell-like cells expressing CD11C will be found up at the very top here. So using this method, we also are able to start dissecting things that would be very hard to do at the transcriptional level. Here, for example, can define that naive B cells, non-switched B cells are primarily to the right, whereas to the left we see the non-switched cells. And using CD45RA antibodies that actually detect a splicing form of CD45RA, we can easily detect which ones are the naive T cells and which ones are the memory T cells not expressing CD45RA. Importantly here, CD45RB is an antibody that detects a glycosylation difference, and we know that that is expressed on memory B cells. You can see that that clearly is more expressed down here on the memory type cells than up here on the naive cells. So this type of definition using these antibodies, CD45RB and RA, would be very hard to do using only the single cell RNA-seq, and ensures that we can define cell population based on the same principles as used for flow cytometry. And finally, we also can then look for the antibody binding. You can see here that the sorted cells, of course, to a very large extent contain N, RBD, or S binding cells, whereas if we take the total amounts of B cells, there will be a very large number of B cells that are not binding to the cells, and also in PBMCs, of course, there will be a large number of cells that will not be binding to our antigens, although rare cells will do that. And combined with actually cloning of antibody chains, we will also be able to detect antibodies binding to these specific sequences, so that we can, for example, look for N-binding antigen specificities and then clone on a single cell level that particular cell that express the N-binding and that antibody, trying to generate monoclonal antibodies against different epitopes. And this is also, of course, continuing work that we are working on at the moment. So my conclusions here would be that it's possible to use single cell RNA-seq together with flow cytometry for analyzing very rare antigen-specific cells. And with now the availability of cell hashing, we can start with even fewer cells per organ or individual by pooling different samples after doing the cell hashing, and in that way actually getting much more data out than we would by just pooling the cells without knowing the donor that for each of the cell types. Also, the Laminar Wash allows us for using antibodies for cytometry, even for very, very few cells, something that would be totally impossible in the absence of the Laminar Wash systems, since all the cells will be lost during actually preparing the cells. And the combination actually allows us to do a much more detailed analysis of antigen-specific immune responses than we would be otherwise. And just for some acknowledgement, I would like to point out INTA and Anneli in Nils, where INTA and Anneli has done most of the work with the B cells and T cells from mouse gut. Patricia and Ruth Palmer's group that has done most of the work on the fly, and it's particularly the single cell work, and Lydia, who has been driving force in the single cell analysis of the COVID patients, together with Hannes and Pauline, and also collaborators here are Anna Lundgren and David Angeletti here in Gothenburg. And also specifically, I would like to point out these current or previous postdocs, Christina Lebrero, Nimitha, and Ekaterina, that has helped us so much with running the single cell RNA-seq and stayed very long nights for getting cells from the sorter and so on to make sure that it would work. So thank you.
Thank you so much, Mats. That was really fascinating talk. And I know we're a little long here, but really appreciate everybody who stuck around. And we did have a couple of questions come in. Before I jump into those, I wanted to just quickly share. So when folks, they signed in, they saw there was a poll really asking what is your lab doing with single cell, essentially. And so I just want to share the results real quick. Of about 60 or so folks who voted, 32% said I do not use single cell genomics. So I just wanted to really just applaud Mats for fantastic explanations of how the single cell works. And also we had 37% of respondents said that they don't really use any of the multiplexing techniques that you discussed here. And I just thought your explanations were spot on. They're excellent. I mean, we're going to be using this recording, I think, for if you want to use it for some of your classes, obviously you could do that. But we really appreciate really digging into the weeds and really explaining how the multiomics and the cell hashing works. I really haven't seen a better explanation myself. And we had 11% said that they use antibodies for multiomics, 5% for cell hashing, and then 16% doing both as you do. So really diverse audience, and so hopefully folks had some good take homes in terms of what they can do with single cell if they're already doing it, to really get some of that extra dimensions. And I think your data really demonstrate what else you can learn besides just the transcriptomics, and also how you can make the most of your samples, how you can save money by doing cell hashing. So really appreciate that. The questions that have come through, and let me just double check that I haven't seen any new ones. I went in and answered them on the as we spoke here, but basically they're about the first couple were about coatings on the plate. So the hydrophobic and the hydrophilic coatings, whether they were stable over time and against certain reagents. So the answer is yes and no. Of course, there are certain reagents that you won't want to use on the hydrophilic label, for instance, acetone. But most of the reagents that you're probably used to using, even methanol, are perfectly fine to use on the laminar wash plates. And the plates themselves are stable for about a year. That's our recommendation. Their shelf life is a year. Dr. Mats, did you have any additional insight on your experience with the plates themselves? Is there some experiment you want to do that you can't because the plates aren't stable?
No, that has not been a problem at all. And I would also like to point out that since we're also using a 96 well washer for a totally different thing in the clinic, and in that case, you can actually use half of the plate and they're stable, so that if you want to sort of reuse non-used wells later on, you don't need to use all the 96 wells in one go. So it's because it's, at least in our case, when we just essentially use PBS for washing the plate, it's okay.
Yeah, no, good point. You can use the plates over the wells that you haven't used. That's a good point. Thanks for bringing that up.
Yeah.
So here's a question for you, Matt. So for human B cells, are the B cells stained with TotalSeq C antibodies after isolation of antigen-specific B cells or before?
Yes, they are. So the point here is that we sort of start with the cell hashing is done before, but then we isolate the cells in different ways. In this case, we isolate the cells as being B cells or being total PBMCs or being the sorted single cell RNA. And then because of the different hashtagging, we mix the cells, and then we have the TotalSeq C antibodies. So usually we prepare the cell hashing in the first step before any further analysis. And then we sort the very rare antigen-specific cells, and then we mix similar numbers of cells from either from the different organs, like I discussed with the mouse work, or from the different isolation protocols for the human work with the TotalSeq antibodies. And then we even mix different individuals, like we discussed for the humans, that there are two different samples. They could actually be from two different individuals or two different time points.
Time points.
Or for
Great.
the so it's no so usually I would recommend though to use the TotalSeq, especially if you're using, they're quite expensive tools. So if you're going to start before isolating your cells, the amount of antibodies needed, for example, to stain a whole mouse spleen would be quite high numbers of antibodies, and that means that it will be quite expensive if you start using 100 different antibodies, I would say. But for if you stain them at the last step, that makes it feasible and possible to use.
Okay, thank you. And I should mention too, so the instrument you're using is our Laminar Wash Mini, and we recently put a new demonstration video on the website, demonstrate how to turn on the Mini, how to use it, how to prep it, and then we actually have a demonstration of a CytoSeq protocol, so similar to TotalSeq, just not branded, that you could follow to see just how straightforward it is to use the Mini system. and how to adapt your standard protocols to using the system if you're interested. And of course, if you want to contact us about setting up a demonstration in your lab, we can of course do that. Last question here from the audience: are there any current labs using this in a BSL 3 or 4 setting? So I can answer that yes, particularly BSL 3. What's really nice about the system, both the Mini that Dr. Bemark is using and also the HT2000, is that they're relatively small. They easily fit comfortably inside a standard biosafety cabinet, and they don't generate aerosols like a standard centrifuge does. So overall, it's a very safe system, particularly for handling infectious samples. And in this era of COVID, basically all human samples should probably be considered infectious. So, Dr. Bemark, do you have anything else to add there in terms of biosafety levels?
No, this was also one concern for us with the COVID samples, of course. And even though nobody has actually described a single case of someone getting it from a bloodborne disease, as far as I know, the levels of RNA is extremely low in blood, if anything at all. Yeah. But I would say that, so when we do the sorting, that has to be done very carefully to follow all the instructions from our facility and so on. But for the actual working with the staining and the washing in the Laminar Wash, we can do that exactly as described here in a normal flow cabinet as a BCL3 or BCL4. No problem.
Great. Thanks. So I have one question. So I really appreciated your explanation of cell hashing, how it works, how we think about the concentration of cells and how many cells per droplet. I really recommend folks to go back and look at that if you need a refresher on how cell hashing works. Just kind of a follow-up with that. When you started using the Laminar Wash system, did you start to see any improvements in doublet or even empty droplet ratios? Because we have customers that have reported that. So I was just curious if you're seeing similar results.
We're working with quite nice and simple cells. Okay. If you've seen a lymphocyte, it doesn't really want to stick to anything else. So it's very easy. So I wouldn't say that we saw a big difference just because, but we haven't looked particularly carefully because we didn't have a big problem even before. Okay. So yeah, usually it's a problem. If it's a problem, then you notice if there's a difference, right? But if it's not a problem for your system, then you won't notice it. But I just wanted, we have customers that have reported working with PBMCs that they do see an improvement in the doublet ratio. So they essentially are getting more data from their samples, and we're trying to figure out exactly how that is coming about. Probably something to do with not pelleting the cells, treating them more gently and having them just settle, and we're able to resuspend them easier. But it's something that we have had customers report. I think that's all the questions for now. So I wanted to say thank you so much, Dr. Bemark, for your time and your fascinating explanations and results here. We are going to be uploading this webinar as soon as it's available to our website. And so all registrants here today will send you a link so you can bypass any gating and go right to watch the webinar again. And we're also going to have a live Q&A for folks more in the European time zone. So it'll be 9 o'clock Mats's time, so in Central Europe on Thursday. So folks, in particular for the poor people in Asia, that if they wanted to attend now, I guess it would be something like
mid-afternoon,
1 or 2 o'clock in the morning.
Yeah, with this webinar, we did kind of, shaft the folks out in the Western United States. Hopefully you came up this morning at 8 o'clock and enjoyed your coffee and breakfast and joined us today. It would be a little bit late at night, at midnight for you on the Q&A. But again, the webinar will be available online. So thank you so much, Mats. I really appreciated working with you on this webinar. And for that, we'll talk to folks later. Thank you very much.
Yes, and I'll see you around on Thursday if you manage to get up at that point.
I'm planning on being there. Thank you, Mats. Take care.